So my name is Michael Kistner, and I am the Director of Operations in the Columbia Stem Cell Initiative Flow Cytometry Core Facility at Columbia University. So the Columbia Stem Cell Initiative Flow Cytometry Core Facility serves the flow cytometry needs of a variety of different researchers across many departments at Columbia University and across many campuses. So we work with researchers whose research area is primarily neuroscience, stem cells, cancer biology, immunology, and bioengineering, among others. And the Columbia Stem Cell Initiative is Columbia's community of stem cell researchers. And the reason why we exist is because stem cell researchers at Columbia are represented by many different departments, and Columbia Stem Cell Initiative brings these researchers together in a community. We have two Sony MA900 Cell Sorters, and these instruments have really changed the way that we operate the core facility. As I mentioned, our kind of philosophy in the core facility is independent access to the instrumentation. And the Sony, because of its ease of use and very robust automation and high performance, has allowed us to train vastly more users than we were able to previously. And some of the main applications where this instrument has really differentiated itself is for users who have samples that are unpredictable. So we have users who you work with clinical samples in a variety of settings, and those samples do not arrive in a timely manner. They're often arrive at users' labs in the late afternoon, and there's no way that they would be able to process these samples and get them on a sorter within kind of typical hours of operation of a core facility. So these particular users are able to, through the ease of use and robustness of the Sony MA900, they are able to prepare their samples, take their time to do that, and then get on the sorter when they're ready, which is often after hours. And these samples can also arrive during the weekend. So these particular users will be able to start up the instruments independently and perform their sorts during the weekend when there's no staff in the lab, which has benefited users and allow them to push their projects forward that would really not be possible otherwise. One of the things we're really excited about is bringing new technology into the core facility. And as working with instrumentation, one of the reasons why we do our job is because we love technology. And one of the most enjoyable things for me is to be able to bring in new instruments and new technology into the lab. And that's the process is very fun to be able to evaluate the technology, be able to bring it in and be able to deploy it to users. It's a challenging process, but it's extremely enjoyable. And even more than that, it serves researchers' needs and helps propel research forward. So one of the technologies that we're currently lacking in the facility is our instruments that are capable of full spectrum detection. So we're looking at full spectrum or spectral sorters and analyzers. We also want to bring in sample preparation instruments or solutions into the core, because that's something that I think is really useful for a core to have, to help users standardize their experiments and to provide another level of service, because we're often involved in the end stage of the experiment, because the sort, by the time the users get to the sort, their sample's already prepared. And if they arrive with a sample which is compromised or prepared improperly or filled with dead cells or debris, that really will compromise the quality of the sort. So being able to bring in sample preparation instruments and tools will help users really generate better data.